genetics, that nightmare is over. Well, true, yeah, it's not over until after the test, but at least, you know, you can take a little break and watch another nightmare, which is metabolism, which is going to be a lot of chemistry. I know you don't like chemistry. Well, most of you. So, in terms of metabolic process, what is metabolic? What is metabolism? Okay, I see where you're going. Yes, it's all chemical reactions, right? It's all about chemistry. Chemistry is foundational there. And this whole chemistry is about how we convert one type of molecules into another and convert chemical energy that can be found in, I don't know, sugars into chemical energy that can be found in ATP. Does that make sense? So it's all about energy conversion, the entire metabolism. Not only this, well, philosophical digression for a second. If you think about life, like general life, all life forms is about converting energy. We convert chemical energy of food into the chemical energy in ATP, which then can be converted into, say, mechanical energy of movement, or chemical energy of things like proteins in the muscles or fat in adipose tissue. Does that make sense? So it's all about energy conversion. Okay? So all metabolic reaction, bless you, all metabolic reactions can be divided into two groups: anabolic and catabolic. So anabolic, right here, are reactions of synthesis. When you take smaller components, smaller building blocks, and put them together into the larger one. We learned about some of the anabolic reactions already. Can you name them? Smaller into bigger. What? Into, how do we call this process? Yes, there you go. Translation is the anabolic reaction, right? So so is transcription, so is replication. Make sense? It's all anabolic reactions. Catabolic is when you break things down. Can you think of any catabolic reactions? The one that you started to learn from like high school. Well, good. In digestion, you break down what? Like big molecules, like proteins or starches, into the smaller building blocks. Okay. One of the simplest catabolic reactions is the ATP hydrolysis. When you break down ATP into ADP and inorganic phosphate, right? Now, interesting thing about catabolic reactions is that often when you break things down, the energy is released. Does that make sense? If you think about it, if you have two smaller molecules and you have to put them together to make a bigger molecule, this will require energy, right? That makes sense? You need to spend some energy to establish the chemical bond. But when you break the chemical bond back, then energy is going to be released. Okay, think about just those are very, very simple concepts. Keep them in mind when we talk about metabolism. Now, again, we're going to talk about the metabolic classifications of microbes. And we can classify them based on two major categories. If you think about any living organism, whether it's a human or simplest bacteria, they need two things. They need energy and they need nutrients, right? They need nutrients to synthesize proteins, DNA, lipids, various types of organic molecules that are needed for metabolism to grow. They need nutrients to grow, and they need energy to use these nutrients to establish chemical bonds between the building blocks of those nutrients, right? And microorganisms can be classified based on the sources of energy and sources of the nutrients. And if you think about the main nutrient, the main building block of any living thing, even non-living thing like viruses, it's organic carbon. Does that make sense? Okay, this is part where chemistry starts to play. Organic carbon, like the carbon in methane or carbon in acetic acid. It's all organic carbon. Organic carbon is in the plastic on top of your computer. Okay, organic carbon is this plastic. Organic carbon is your skin. Organic carbon is polyethylene sack that you carry your groceries from the store. It's all organic. Okay, so based on the source of energy, organisms can be classified into the chemo and phototrophs. Does that make sense? So phototrophs, where do they get energy from? Light. Chemotrophs, where do they take energy? Chemicals, yes, chemicals. Okay? Does that make sense? Now, auto versus heterotrophs. So when we refer to heterotrophs and autotrophs, we now refer to the source of carbon. All heterotrophs take carbon that is already organic. So where can they source it? Heterotrophs. Where can you find organic carbon in the environment? Nope, it's not organic. That's inorganic. CO2 is inorganic carbon. We'll get to that. Yes. Excellent. How do we call guys that eat other organisms? Well, like, we have more offensive word for them. Parasites. Like living organisms, right? Can we take inorganic carbon from the dead organisms? Like if I'm dead, my carbon, is it still organic? Oh yeah, whole foods grade. So they can take it from me, right? It's also heterotrophic. So trees, plants, dead trees, dead plants, dead organisms, and so on. Heterotrophs take carbon from organic sources, autotrophs from inorganic sources. So Hayden already mentioned one of the major inorganic sources of carbon, CO2 in the air. Let's talk about organisms that surround us. Those guys that still don't have leaves. Okay? Where do they take energy, the plants? What? Sunlight. Yep, they take the energy from sunlight. So based on the energy source, they are phototrophs. Correct? Where do they take carbon from? Air, CO2. So they photo-autotrophs because they use sunlight as an energy, photo, and inorganic carbon as the source for carbon for growth. So photo-autotrophs. Not only plants, all photosynthesizing microorganisms that can fix carbon. In fact, if you would take, if you would write the generic equation, C is the carbon, inorganic. Some organisms, like plants, they can convert inorganic carbon in the air into the organic carbon, like the one in starch. That makes sense? So this process is often referred to as carbon fixation. Does that make sense? Okay? So that's photo-autotrophs. Can there be photo-heterotrophs? Absolutely, yes. There are microbes that use light as a source of energy, but they take carbon from the environment, from the organic sources, usually from decomposing organic matter. Those are called photoheterotrophs. Different types of bacteria. Okay, they lack clinical significance. We're not going to talk about them. They're extremely interesting, but we're not going to talk about them too much. Now, Chemo Heterotrophs. So chemo means the source of energy is chemical. What is the most common source of energy, chemical source of energy? Which class of nutritional molecules? What we humans use as the to-go source of energy. Huh? What? See again? Yeah, glucose, carbohydrates, glucose. Okay? I mean, not necessarily only glucose. We can successfully metabolize lipids and proteins, and so do bacteria. Bacteria can do that too. You know, they can use glucose, or they can switch to fats, or they can switch to proteins. Okay? So it's chemo. Hetero means that the carbon that this microbes or organisms use, carbon comes from organic sources. Okay? Where do we take our organic carbon? Can we fix CO2? No, we can't. We have to eat organic matter, right? We eat plants, we eat animals. So we are Chemo Heterotrophs. And most of the bacteria and most, I would say not even, yeah, probably most fungi are Chemo Heterotrophs. Okay. Does that make sense? Protozoa, most of the organisms that we're going to talk about are Chemo Heterotrophs. Another interesting, extremely interesting group of organisms, Chemo Autotrophs. So they use chemicals as the source of energy. Does that make sense? But it's not our conventional chemicals like glucose. They use usually inorganic chemicals. They can what it's going to be? They can oxidize things like sulfur. They can oxidize hydrogen. They can oxidize nitrogen. Okay? In order to gain energy from those inorganic sources. And they also are capable of fixing carbon from inorganic sources too. whether it's carbon dioxide or it can be carbonate ion. So in terms of inorganic sources of carbon, there are two major sources. It's CO2, okay, or carbonate ion, right? Those are inorganic sources, yes. Does that make sense? So we've got here 4 groups finally, 4 groups of microorganisms. And these groups will include pretty much every microorganism on Earth, right? Most of the microorganisms, especially the ones of the clinical significance, are going to be Chemo Heterotrophs. But microbial organisms in the environments can successfully use light, the source being phototrophs, or inorganic chemicals being, again, chemotrophs, or they can obtain carbon from inorganic sources, being autotrophs. By combining those strategies for energy acquisition and carbon acquisition, we can classify each organism into one of those groups. Does that make sense? You will absolutely get the question, such and such microbe uses blah 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 as the source of carbon and uses blah 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 as the source of energy. What it is. I'm not gonna make it, again, extremely hard, but you have to quickly recognize, chemical versus light, organic versus inorganic. Okay? Now, when we talk about metabolism, when we talk about energy sources, what is energy? No, that's a bad question. Energy conversions in the living organism. We started this lecture from this topic. What is established and broken down during the energy conversion? Chemical bonds. Chemical bonds, right? They are established, broken down. Now, chemical bonds consist, from the chemistry standpoint and even physics standpoint, consist of which elementary particles? You have a hydrogen atom and another hydrogen atom, and they form hydrogen molecule. What the bond between atoms consists of? Which elementary particles? Say what? Electrons. Very good. Electrons. So if you think about this, if every bond consists of electrons, when you break it down, you have kind of electrons back and forth. You can move electrons from one molecule to another, from one atom to another. So essentially, the energy conversions in the living creatures, in living organisms, can be represented as the flow of electrons. Does that make sense? That is why when we talk about energy sources, we usually talk about stripping electrons from certain molecule. Those electrons that were stripped become high energy electrons. Okay? So this is this one sentence explanation. Those high energy electrons, as they go from one molecule to another, it's an energy source. Okay? And you can think about it this way. You have let's talk about Chemo Heterotrophs, okay? JCD mentioned what is the major source of energy in chemotrophs? Glucose, right? Molecule of glucose. So I have molecule of glucose, you strip it of the electrons. And then electrons can't just flow around forever, so they have to go to some other molecule. Any idea what this molecule is going to be? What's going to be the acceptor of it? So donor of electrons is glucose. What's going to be the acceptor? Huh? No, but you're in the right track, I think. Not hydrogen. The final electron acceptor in the respiration. Say again? Oxygen. Very good. So we strip electrons from glucose, and those electrons end up on the oxygen. We can essentially think about it as glucose being right here, oxygen being right here, electrons rolling down the hill, and as they roll down the hill, okay, that is energy that they produce. Like when you take a gigantic ball and roll it down the hill, it's going to get certain energy at the bottom. Does that make sense? Okay? You see the point here? So we have to transfer electrons from one molecule to another to acquire their energy. How do we call reactions in which electrons move between two molecules? They're right there. You can just read oxidation and reduction. For the short, we call them redox. Right? Reduction and oxidation. Okay? Does that make sense so far? Okay, you follow. So this is the ultimate scheme. Okay? Now, you cannot have one without another. These are so-called coupled reactions. If something is getting oxidized, it means that we strip this molecule from electrons. That make sense? If something is getting reduced, it acquires electrons. Okay? Now, since I am tethered, I won't be able to demonstrate it successfully, but imagine that this lamp is a molecule, okay? And I'm another molecule, and this is my electron. So if I'm being oxidized by this lamp, I lose my electron, and the lamp gains it. Does that make sense? I can oxidize this lamp and get my electron back. Okay? When lamp oxidizes me, and I am losing my electron, I am reducing the lamp. I'm a reducing agent. Does that make sense? I'm a reductant, and lamp is oxidant. So reducing agent, another name that you can give it is reductant. And for oxidizing agent, it's going to be oxidant. Does that make sense? Does it? Okay. Does? Okay. So, in the end, you see, this is oxidized, and this is reduced. So, going back to our example with the glucose as the electron donor, and oxygen as the electron acceptor. In this reaction, in the reaction of respiration, which is this. I'm not putting any coefficients now. Okay. Now, oxygen is the electron acceptor. What else? How else can we call oxygen? Oxidant. And glucose? Reductant, yes. So in this reaction, glucose is Oxidized or reduced? Oxidized. Glucose is oxidized because we take electrons from glucose. And oxygen is reduced because it gets electrons. It's what's passive tense, passive voice, okay? Oxygen is reduced because it gains electrons. Now, what oxygen does to glucose? It oxidizes it. And glucose reduces oxygen. Okay? Please be familiar with the terminology here. Okay? Now, electrons, can they travel around the cell just by themselves? No? Why? Oh, come on. Hypothesize. Why they can't travel? Why electrons just can't move from one, like you have glucose in this corner of the cell and oxygen in that corner of the cell, and electron just sprints its way across the entire cell. A what? No, I mean, let's say you have some kind of way to strip the electron from glucose, and free electron will just make its way across the cell to another corner of it. Huh? It is charged. Okay, fair enough, yes. That's one obstacle. It's probably the most reactive particle that you can think of. Electrons will wreak havoc everywhere. If you've heard about different types of radiation like alpha, beta, gamma, beta radiation is electrons, and it's pretty bad. So electrons are damaging. They can destroy molecules. So they can just, freely roam around the cell. They have to be carried. Okay? So there are special molecules called electron carriers. One of them is shown here. It's NAD. Nicotine, amide. So that's nicotinamide. Adenine dinucleotide. Okay? You don't have to know what acronym stands for, but what I want you to know is, what I want you to understand is that NAD, okay, can get a couple of electrons, okay. Since electrons carry negative charge, it also has to get a couple of hydrogen ions to balance for that negative charge, and then NAD, when it gets electrons, is it oxidized or reduced? Reduced. So reduced form of NAD is NADH. Okay. NADH then carries those electrons to wherever they are needed. Does that make sense? They're like taxi that move electrons from one part of the cell to another. Does that make sense? Another electron carrier is FADH. For FADH, the reaction will be, plus electron plus hydrogen atom is going to be FADH2. Okay, that's the FADH2 is the reduced form of FADH. Does that make sense? Now, what is the sort of a final step for the electrons? Why do we want this reaction to happen? Because this reaction will produce what? When you burn glucose, essentially burning a glucose. Energy, very good. And this energy then can be converted into ATP. Now, philosophical question again. sort of philosophical. Why ATP? Why we cannot just burn glucose? This burning of glucose will produce energy. Why can't we use this energy for intracellular processes? Why ATP? It's not a simple question. Think about this word. Does anyone do any craft? Like any I don't know. I have two left hands, so I have no trade that I can offer to people, but can anybody offer a trade? I don't know, plumbing, HVAC, sewing, something. I hope somebody can, otherwise we're all screwed. Okay? So, now think about this. You bake bread, you do plumbing, you do something else. Okay? Now, of course, we can try and figure out, if I fix your car, you're going to give me 5 loaves of bread. Or 55, which I don't know what to do with, then I will have to use 50 loaves of bread to get something else, you know. So this natural exchange doesn't really work terribly well. Huh? Bingo. ATP, universal. Bacteria don't really care about other cells, but what's important, ATP is universal energy source for all cellular processes. Because think about this. You have protein synthesis. It requires energy. Of course, technically you can think of some enzymatic system that will directly use glucose to fuel up translation. Does that make sense? What if you run out of glucose? You're kind of screwed. You gotta have a backup system specifically for the translation to use, I don't know, lipids, right? Instead, you have one dedicated enzymatic system that produces ATP. And then you have a universal way to extract energy from ATP molecules, whether it is for transcription, translation, or replication, or something else. Does that make sense? You have those molecules that are called ATPases and GTPases. Does that make sense? So instead of doing the natural exchange, you just pay with money. So ATP is money, energy money of the cell. Right? So that energy of electrons moving from the reducing agent to the oxidizing agent, from say glucose to oxygen, that energy is essentially stored in the gamma and beta phosphate bonds. So this is gamma, this is beta, this is alpha. Okay? So these two bonds, when they are broken, question? Yeah, I don't mind. Any questions? So when those bonds are broken, energy is released. The highest amount of energy, of course, is released when you break down the gamma phosphate bond. That's why we have this ADP plus phosphate. Does that make sense? So if you look at this, the reaction in which ATP is being synthesized consumes energy and stores it in the form of phosphoester bonds. The reaction in which those bonds, the phosphate bonds, are broken down, energy is released and can be used for other cellular processes. Does that make sense? Okay, so far what we have figured is that different energy sources, chemical or physical like light, can provide energy that is converted into the energy of the chemical bond in the ATP. And essentially it's a movement of electrons. You will see it even in what's the word? Photosynthesis. It's about electrons moving. Okay? Now, how do we call reactions? that produce energy or acquire energy. Reactions that release energy are called exergonic. So ergo, energy, X from exit, energy exits from reaction. And that's my way for memorizing it. So catabolic reactions, reactions of degradation, are mostly exergonic, right? For instance, when ATP is broken down to ADP and inorganic phosphate, energy is released, and by definition, this is catabolic reaction. Does that make sense? Again, glucose. When glucose is oxidized, either in the process of glycolysis or glycolysis plus Krebs cycle plus electron transport chain, oxidative phosphorylation, all those reactions. Look, you start with a molecule of glucose and you end up with CO2 and water. It's definitely catabolic, okay? And it also produces energy, so it's exergonic. On the other hand, process in which the ATP is made, synthesis of ATP, okay? It's endergonic and anabolic, okay? A reaction of synthesis. When organisms, say a bacteria produces some sort of a polysaccharide, or bacteria produces protein from individual amino acids, that's anabolic reaction that requires energy, so it's endergonic, okay? So that's not only endergonic, that's anabolic. Does that make sense? Now if you, put it all together in the cell, in the most cases you can see that reactions of catabolism, reactions of degradation, will also release energy. And that energy will be later used in the reactions of anabolism, which are endergonic. Does that make sense? Okay. It's all about energy flow. Can I burn this piece of paper? I'm not asking may I, I'm asking can I? If I will start it on fire, I don't want to because the fire extinguisher is far and water is far, but if I'll start it on fire, even now it's also fire, and I don't have a lighter, well, probably can reach for matches. Matches, here. Can the match burn in the air? I can prove right now it can burn in the air, and I don't need any energy when it burns, right? It burns by itself. True? You want to see? Sure. You know, I don't provide any energy. It burns by itself, right? That make sense? Come on. It burns by itself. Oxygen plus whatever it is in the match gives you a flame. Oxygen, whatever's in there. Does it burn? Yes, we need something to start the process. So what do I have to do? And friction produces heat, okay? So I need to heat it up. Once I heat it up, it goes by itself, right? Now think about this. When you build a bonfire, all you need to do is just a little bit of a heat to start it, but then it goes by itself and you just throw firewood in and it produces an enormous amount of energy, but you really have that start. Does that make sense? The start is called activation energy. Some reactions need it, some reactions don't. One of the best examples for activation energy is the reaction between hydrogen and oxygen. What will be produced as the result? Water. Very good. Now imagine for a second that you have hydrogen and oxygen and you mix them together in some kind of a vessel. What's going to happen? Hydrogen, oxygen, you mix them together, what's going to happen? I'm going to tell you nothing. You can leave them there. I think it takes about like 100 years for the first drops of water to appear. So yes, reaction goes by itself. Slowly, like unbelievably slowly. Okay, but you put down the light, the match, into this vessel with oxygen and hydrogen, and you're a toast. I mean, physically a toast. It's going to explode right away. Okay, so this is called activation energy. That little bump in the beginning of reaction that you have to overcome to produce, to start the reaction. Okay, it's called activation energy. Right? Does that make sense? Now, for pretty much all the chemical reactions in the cell, activation energy exists, and it's really fairly high, and the reactions cannot go by themselves, or if they can, they will go extremely slow. Right? Now, a cell cannot afford slow reactions. The chemicals, the molecules known as enzymes, okay, they facilitate the reactions so that they go faster. That makes sense? Now let's go back to the reaction that I put right here, this one. If you have unlimited amount of time, eventually all molecules of hydrogen and oxygen will form water. It may take you, I don't know, 1,000,000 years. Okay? Now if you had a match, in quite literally split second, all molecules of hydrogen and oxygen will combine to form water as well. Does that make sense? Because the activation energy, you jumped over it using the burning match. Okay? Now, what I want to highlight in this example is that in both cases, you have the same yield of the product. Does that make sense? All hydrogen and oxygen molecules combine to form water, all of them. But in one case it took 1,000,000 years, in another case it took a second. But all of them eventually were used in the reaction. So yield is the same. The rate is different. That's the feature of enzyme. Enzyme doesn't alter the ratio of the products. Does that make sense? It doesn't alter the ratio of the products. It doesn't alter the yield. It alters the rate of the reaction. Does that make sense? Okay? Increasing rate, but not ratio. Alright? We're good about that's extremely important feature of the enzymes. Okay? Now, pretty much all the enzymes are proteins, except for some that participate in the translation. The peptidyl transferase is the RNA, and splicing. The concept of the enzymatic reaction is very simple. Substrate of some sort binds to the active site. Whatever is in the binding site, usually it's a bunch of amino acids, amino acid residues that comprise the enzyme. They convert substrate into the product. Okay? And you now have free enzyme and you have products. What happens to the enzyme during the reaction? Okay, Does anything happen to the enzyme by the end of the reaction? Nothing, okay? Enzyme stays the same. Enzyme is not chemically used in the reaction. It's not chemically modified. Does that make sense? That's why it is called a catalyst. Okay, so this reaction that you see on this picture, you can write it down in a way that you have a substrate. Okay, substrate is converted into the product, and enzyme facilitates this reaction. Are we cool? Now, enzymes are extremely specific. So there are different models. One that is represented here is called induced feed. I know that since I know that, I will tell you that it is induced feed. If you would look at the shape of the binding site before and after binding the substrate, you can see the slight differences, right? So substrate changes the binding site, the active site of the enzyme, slightly. Does that make sense? But pretty much what you can think of, every substrate is very specifically recognized by a particular enzyme. Think about the digestive enzymes, bless you. Enzyme like a sucrose will facilitate the breakdown of regular sugar into the molecule of glucose and fructose. Enzyme such as lactose will facilitate the breakdown of lactase will facilitate the breakdown of lactose into the glucose and galactose. So sugar is similar in structure, fairly similar, but the enzymes are capable of, bless you, distinguishing them and specifically bind them and, do things. Does that make sense? They are extremely specific, right? Now, each enzyme has optimal conditions for function. Enzymes that normally function, say, I don't know, RNA polymerase that does transcription, what is the optimal pH for it? What do you think? RNA polymerase from the human cell. What's the optimal pH? Ballpark. 7.4, okay, 7-ish, neutral, okay? The enzyme that, pepsin, breaks down proteins in the stomach, what's the optimal pH? 3, 4, acidic, yes. Does that make sense? So, each enzyme has the optimal pH. Now, you have to understand that if you would plot the enzyme activity, we'll call it A, and on the x-axis we will put some sort of environmental condition, it looks like this, okay? Whether its environmental condition is pH or temperature, okay? So enzyme will have the best condition, but it will also function, although not as good, at different conditions. Does that make sense? So temperature, pH, ionic strength, concentration of substrate, concentration of product. Okay, so all these factors may contribute to the enzyme regulation. Does that make sense? Let's summarize I'm going to summarize the enzyme, pay a little bit more attention to that graph, and then we're going to take a break. All right? So, first, enzymes. Do they change rate or ratio? Rate. How do they change it? They increase, they speed it up. When you add 1 microgram of the enzyme, which is like ridiculously huge, just unimaginably high. But anyway, you add 1 microgram of the enzyme in the beginning of reaction. When you're done, how much enzyme ideally you have? Same amount. They are not chemically modified during the reaction. Okay? What about specificity? Extremely specific. Recognize unique substrate. Chemically, What are they? Most of them. Proteins. What did you say? It's okay. They catalyst, and they proteins, okay? They proteins. Most of them are proteins. All of them are catalysts. Now, how do we call chemical that is converted? That is substrate. Enzyme converts substrate into the product. And substrate binds to what part of the enzyme? How do we call that? Binding site. Active site. Okay, that's kind of a wrap-up of terminology. Does that make sense? Now, how can we tell that something is the enzyme? It ends with ase. Like lactase, polymerase, ends with ase. That's enzyme. Right? That makes sense? Good. Now let's take a glance at this graph right here. So this graph represents some generic reaction, okay? When you have the X into Y reaction. Does that make sense? Okay? So X is right here, Y is right here. So when X is converted into Y, we don't really know what those chemicals are, we're not really interested. This is the amount of energy, the green arrow, that you have to spend to get over that hump to initiate the reaction. Does that make sense? Okay? Now when you convert Y into X, this is the amount of energy that you have to spend to convert Y into X. Does that make sense? In both cases, enzyme reduces that activation energy. You got it? Now you have to appreciate that it reduces activation energy for both forward and reverse reactions. It doesn't distinguish between those. Alright? Now, last thing that I want to mention. Delta H is called enthalpy of the reaction. Enthalpy is the fancy word which means the energy that is released or required for the reaction to go. Now reaction X to Y, will it require energy or will it release energy? Require. Why? Say again? Well, that's one thing, but in terms of the, how to say, like you burn paper, you have energy release, right? Although you need some energy to start it up. Okay, so that's going to be exergonic reaction. Now this reaction, let me make it more specific. This reaction X into Y, is it endergonic or exergonic? Huh? Endergonic. Why? Not only because of the activation energy. Activation energy will kind of compensate itself. Why else? Can you tell me which part of the graph allows you to make such conclusion? Products have higher energy than reactants, right? You see that difference? That difference? Enzyme doesn't change that. It doesn't change the difference in energy levels between the products and reactants. Does that make sense? So this tells you, okay, we're going to use energy to make the reaction run through. But if we would look at the reverse reaction, Y to X, now this one will go with energy being released. Does that make sense? Because you kind of climb from the hill. Okay? Good. Let's take a break, and we're going to do some microscopy after the break.